Four, three, two, one. Shruti, over to you.
Good evening and welcome to Project Step 1 webinar, and I am Shruti Bharadwaj. It's my pleasure to introduce Project Step 1, a collective of 5,000 plus doctors and multiple startups from all across India have come together to support the government in this time of crisis through teleconsultation and technology. Our mission is to help government triage, identify, locate COVID-19 suspects quickly so that we can prevent the spread of the virus. Today, this webinar, I welcome Dr. Shashidhara MK, International Medical Graduate Tutor, Stockport NHS Foundation Trust, Deputy Lead for Quality, Global Training and Education Center, Honorary Senior Lecturer, Edge Hill University, currently involved with supporting nurses and international doctors through the Global Education and Training Center at Wigan Wrightington Lee NHS Foundation Trust. I also welcome Professor David Baxter, Consultant in Public Health for Stockport, Founder and Director of Clinical Vaccinology, and Dr. Shiv Shankar, President-elect Indian Orthopedic Association. Welcome you all. Doctor, given the current situation, we know that the best exit to the COVID-19 crisis is through a vaccine. So the question on every individual's mind is, when will the coronavirus vaccine be ready? We are all living in a state of anxiety and uncertainty. We can't go out even because of the fear of catching the infection or infecting our families. At this stage, the only thing that can restore our lives to normalcy is a vaccine. Can you please share the hope of ray for this pandemic and also introduce the other doctors in brief? Over to you, Dr. Shashidhara.
Thank you. Thank you, Shruti. Let me just bring my screen. Right. Shruti, thank you very much for that. And thank you for step one and the Abhineva team to give us opportunity for allowing us to share our experiences. I'm here on the behalf of Global Training and Education Center, which is part of the Global Medical Program, which is the second most common training program in UK for international medical graduates. So first and foremost thing, I want to actually thank my friend, my classmate, and the orthopedic surgeon, Dr. Shiv Shankar. Dr. Shiv Shankar has agreed to chair this session. Dr. Shiv Shankar is an orthopedic surgeon in Stockport. He is well known for nailing various technology of nailing in orthopedic surgery across India. And he is the current president-elect of the Indian Orthopedic Association. Shiv Shankar, welcome.
Thank you very much, Shashidhar.
Okay. As I said before, the Global Training Education Center does support whatever is happening across the COVID situation in India. I know things are becoming worse. But what we are trying to do is to share common learning between UK and India, try and help each other in terms of tackling this pandemic. So for this reason, I actually requested Professor David Baxter to be able to do a session on coronavirus vaccines. I know Dr. Baxter for some time. David is the current Director of Medical Education in Stockport. In actual fact, I'm very proud to say that he's also my mentor. He is very keen for, and actually sets up standard in terms of delivering undergraduate and postgraduate medical education in Stockport. He's deeply involved with medical education using simulation. In fact, David is a ATLS instructor. The reason why I asked David is David is a consultant in communicable diseases control for more than 30 years. In fact, he's the founder of the clinical vaccinology. He's just finished 32nd clinical vaccinology conference and is still going. And David also is a consultant for public health medicine, and he is in charge of public health in Stockport. And David has been involved with several vaccine trials. if you Google David's name, you will see his name for MenB cluster difficile. Currently is involved with coronavirus SARS-2 vaccines. David is involved with health protection, epidemiology of infectious diseases, and like I said, he is very much involved with training healthcare workers, everybody in national immunization through his national immunization conference and vaccinology workshops across Greater Manchester. And over to you, David.
Okay, so good evening. Can I just say thank you, Dr. Shushankar, for inviting me. Shashi, thanks for that really nice introduction. And Shruti, thanks for helping me set things up beforehand. And since you set things up for me beforehand, I'll try and answer your question. So you want to know when there's going to be a vaccine for coronavirus? Well, you've got the biggest vaccine manufacturer in the world in the Serum Institute of India, and they're planning to deliver, I think, more than 1 million doses initially of a coronavirus vaccine at the end of this year. So, and the other interesting thing about Serum Institute, the Serum Institute of India, it's engaged in probably producing 4 or 5 of the candidate vaccines. So it's going to be a major player in delivering coronavirus vaccines. Okay, so hopefully I've answered your question. Yeah.
Yeah.
Okay, so let's see if I can get my head around this technology. Okay. Right, I hope you can all see that.
Yes, sir.
And you can hear it. Excellent, great. So Shashi asked me to talk about coronavirus vaccines. And I think one of the reasons is because, as he said, we are involved in Stockport in vaccine studies around coronavirus vaccines. So we're expecting to do a phase 3 study, which is basically a study just before you license the vaccine. It's been through animal work. It's been through the first 2 phases of a vaccine study, which is basically, is it safe and does it provide an immune response? It's been through both of those, and several of the vaccines are looking pretty promising, and they're now moving on to what are called phase 3 studies, much bigger studies, these involving thousands and thousands of people, to see if the vaccine is protective. We're hoping to start our first one in September or October this year. There are a number of bigger studies coming to the UK that we're going to be involved in, and I know that the Serum Institute of India is also planning some very, very big studies in India of coronavirus vaccines. We're at the planning stage for this, and as I say, we hope to start sometime in September. Now, again, I'll answer your question, Shruti. What's the first vaccine that's likely to come? Well, according to the CEO of the Serum Institute of India, they're hoping to have about 1 million doses of the AstraZeneca Oxford vaccine. And I'll talk to you about that at the end. They're hoping to have about 1 million doses of that available at the end of the year. So just a little bit of a background. I'll give you a little bit about a background about vaccines, and I'll give you a little bit of background about the disease, and then talk about the different types of coronavirus vaccine. So this is hopefully a short video, very short.
If all of this are okay, we try to ask the mothers about what She's complaining first. And in this case, the mother is complaining of the weakness of the lower limbs. At the first sight, we see that the child can use his upper limbs, but the lower limbs cannot move well. It's dragging. In this case,
Okay, that's polio, folks. And you can see that child has got an inability to move both of his lower limbs. And this is due to poliovirus infection. When I was much younger, there were thousands of cases of polio. We're now getting to a stage where there are relatively few, perhaps in the 10s, 20s, 30s of cases of polio. And we are getting to a stage where polio will be eradicated shortly, perhaps in the next 3, 5, 7 years, something like that. And that's largely due to the effectiveness of a vaccine. And there are two vaccines we use. We use a live vaccine, which is the Sabin vaccine, and we use a killed vaccine, which is Salk. Now, the first polio vaccine was tried about 1910, but it wasn't until about 1954 that we had an effective vaccine. Now, one of the things I'm just trying to demonstrate with that, apart from how effective the vaccine is in terms of reducing numbers, but actually the fact it took more than 40 years to get to a stage where we had an effective vaccine. Let's move on.
The final case is a tragic one of a 6-year-old boy who was admitted to San Lazaro Hospital the previous night with gross respiratory obstruction, which you can see clearly requires urgent action.
That's enough. That's diphtheria. And the child has got a very thick membrane at the top of his throat, and he can't breathe properly. And you can see that it makes breathing very, very difficult. Unfortunately, this child died 6 hours after this picture was taken. We rarely see diphtheria now. It's certainly in the UK, I think we've had 1 death in about the last 6 years. And that's purely and simply due to the availability of an effective vaccine. So again, I mean, just look at this. We had 60,000 cases in 1914. In the last 10 years, we've had 1 case. Okay, and we use a vaccine called a toxoid vaccine. Basically, diphtheria releases a poison or a toxin, and it can cause upper respiratory obstruction. You can change the toxin and make it into a vaccine that is highly effective. This is the last one, and this is going to be a little bit longer. This is about influenza, and it's going to be a couple of minutes long. Just listen towards the end about the description of the disease in the troops, and then just think about SARS coronavirus 2.
Nowhere to drain. Men's feet rotted in their boots, a condition called trench foot. Dead bodies lay decomposing in no man's land between the two armies. When it rained, water would wash excrement, decomposing body parts, and filth down into the trenches where the men lived. Constant bombing churned up the dirt and turned the ground into a sloppy swamp. The mud became so bad that in some places it was like quicksand. Planks were laid down for the men to walk on. There were cases of horses, pack mules, and even soldiers slipping off the walkways and drowning in the mud before they could struggle out of their heavy gear. By the end of the war, over 9 million combatants and 7 million civilians would die in the war. Thus, it's not surprising that when a virulent new virus arrived, it became an even more deadly foe than the enemy soldiers. At some point in the summer of 1918, the flu mutated to become more deadly. It gave some people the normal symptoms of fever, chills, nausea, aches, and diarrhea, and many got sick, then recovered like a normal flu. Some were sick longer or died after catching a secondary infection, such as pneumonia. However, many people became violently ill with strange symptoms. In fact, in the July 1918 edition of the medical journal The Lancet, doctors argued that this strange epidemic couldn't be the flu because the symptoms didn't fit. Italian doctors argued the same. In some people, Spanish flu caused fevers that were so high people hallucinated. Some writhed in agonized muscle pain so bad that doctors thought they had dengue, also called breakbone fever. It made some people temporarily or even permanently blind, deaf, or paralyzed. Some lost the ability to smell. Some had strong vertigo and would fall over if they tried to walk. Extreme ear infections developed very quickly, going from the first pain to the eardrums rupturing within only a few hours. Some had terrible headaches and double vision. Severe mucus excretions and inflammation made it hard for victims to breathe. Some people coughed so hard they tore their abdominal muscles. Doctors doing autopsies saw lungs so damaged that they resembled those of people who died from poisonous gas in the war. Some people developed a symptom most physicians had never seen before. Tiny puffs of air would leak out from tears in the lungs and get trapped beneath the skin, puffing up in little pockets all over their bodies. When they moved, the pockets would crackle like a bowl of Rice Krispies, according to one nurse. Some people developed hemorrhagic fever, which, like Ebola, causes its victims to bleed. An Army report described the flu as a rapidly escalating infection and lungs choked with blood, fatal in from 24 to 48 hours. Some people bled from their nose, their ears, and their eyes. Some people with the 1918 flu became so oxygen-starved they began to turn blue, or even looked black, a condition called cyanosis. People reportedly turned so dark that it was difficult to distinguish white people from people of color. For this reason, the flu picked up the nickname the Blue Death, and many wondered if the Black Death had returned. When people began turning blue, doctors knew they wouldn't survive more than a few hours. The flu was also terrifying because it could kill so quickly. Many victims died within a day or two, or even hours of showing their first symptoms. According to a story recounted in the book The Great Influenza, a man in Cape Town, South Africa boarded a streetcar just as a conductor died. During the 3-mile ride to his house, 6 more people on the streetcar died. When the driver died, he got off the streetcar and walked home.
Okay, that's influenza. And
nowhere to drain. Men's feet rotted in their boots.
That's influenza, initially thought to be due to a bacteria, and we subsequently identified viruses in 1933 that caused it, and we started to use a vaccine within about 20 years of that, and quite an effective vaccine. So let's move on. What I've tried to do initially is just show you how effective vaccines can be in relation to polio, diphtheria, and hopefully I've shown the impact that a disease like influenza can have, and that we now have relatively effective vaccines against that. So this is Wuhan. This is the market where it's thought that Covid-19 started. It probably didn't actually. Probably where it started was in southern China, south of this food market, in about September. The cases then came to notice at the food market in December, where the disease was amplified. But it probably started a little bit earlier, and as I say, in South China. So this is the pandemic. You can see that there were cases reported towards the middle or late December, and people were presenting with atypical pneumonia. That means when you look for an organism, you don't find it. You don't find the pneumococcus that you would normally find. You don't find viruses like flu that you would normally find. They couldn't find anything until early January, when they found a coronavirus. Coronavirus is well known. They're RNA viruses. They've been around, identified since the mid-1960s. And in fact, most of the people watching this will have had coronavirus infections. but they're generally mild. They cause a common cold. But this one has been different. So this is the outbreak in China, and you can see how it's increased substantially over a relatively short 5-month period. And these are just the different cases. These are cases in different parts of the world. So this is you're blue here. This is Southeast Asia. And I think of Southeast Asia, India has the majority of cases. I'm hopeful. This is where I live. I live in Europe, and I'm hopeful that what we're seeing here is a reduction now in cases. Of course, the big concern is, will there be future spread? And almost certainly there will be, unless, as Shruti indicated earlier, unless we have a vaccine that can provide herd immunity. But we'll talk about that. This is where the majority of cases are at the moment. And as you can see, it's in the Americas, it's in Brazil, it's in Southeast Asia. I think it's in India is having a tricky time at this moment. So this is the global situation. We've had 10 million cases with 500,000 deaths. Absolutely huge impact of a new virus. Okay, well, and it is a new virus. As I say, coronaviruses have been known since the mid-60s. 4 of them cause the common cold, but 3 of them have had quite an impact. The common cold viruses just affect the upper respiratory tract, but 3, MERS, SARS, and COVID-19 can also affect the lower respiratory tract, causing pneumonia and other conditions. And that's why they've had such a much higher morbidity and mortality. So this is the virus. You can see it's a small round virus. It's called an RNA virus. That means the recipe that the virus carries to produce more copies of itself is coded in something called ribonucleic acid, or RNA. And this is one of the biggest RNA viruses. You can't see it particularly well, but it's got a sort of fuzzy coat on the outside. And this is a cartoon diagram of it. And these are the fuzzy bits on the outside. These are called spike proteins, or spike glycoproteins. And basically, they're the key that the virus uses to attach to cells. Viruses replicate, and that's their purpose in life, is to replicate. And the only way they can replicate is to get into, in this case, a human cell. And once inside the human cell, they release their genetic material, or the recipe, and that recipe tells the cell how to make more viruses. And as the viruses are made and released, they can then transmit to other cells, or they can transmit to other people. And when the virus is released, it kills the cell. Now, coronaviruses are divided into 4 categories: alpha, beta, gamma, and delta. And the 3 viruses I've just talked about are the MERS and SARS, and there's SARS, and SARS or COVID-19 are all beta coronaviruses. They're in this particular group. How is it spread? Well, we all know that it's spread by the respiratory route. It's spread by coughing, and you can see there are these large particles, and if these particles hit you on the mucous membrane, eyes, mouth, nasal membranes, they transmit the infection. So if this chap is infected with SARS coronavirus 2, coughs out, these bigger particles hit you, they're laden with virus, and it spreads that way. And that's why the importance of social distancing, and also the importance of wearing masks, which is increasingly now recommended. There are some very small particles here that you can't see. They're so small, they remain suspended in air, and it's thought that they can also transmit virus, but probably not as efficiently as these bigger particles. The other problem with these bigger particles is they settle on surfaces and they can persist for 24, 48, 72, 96 hours, depending on the surface. So I think, for example, cardboard is about 24 hours and steel is about 3 days. And again, this is why it's so important that environmental hygiene cleaning is carried out, because when these big particles settle on surfaces, if you come along, touch it and rub your face, then you can transmit the infection. Right, so we're now getting onto the vaccine bit. This is thought to be the how the virus replicates in human beings. This is how the virus multiplies. So you inhale the virus, so it gets into the upper airways, and this is a respiratory cell. And on the surface of cells are locks. These are little locks that, if you have the right key, you can use to get into the cell. Now, this lock is really important for a drug system called renin-angiotensin system, which is related to blood pressure. But over time, and I have no idea how it's happened, this SARS coronavirus 2 has developed a receptor that enables it to attach to this, it's developed a key that it enables it to attach to this ACE2 receptor. Having attached to it, it gets taken into a cell. The recipe, or the genes, the RNA genes are transcribed, and the virus then makes more particles. The particles are then joined with other proteins of the virus, and you make a new virus. And this virus then buds off and attaches to another person. Now, this process of replication can be associated with killing of the cell, and that can then lead to a number of other complications that we'll briefly mention. Okay, does that make sense? So the virus, you inhale it, it attaches through its key, through this this spike key, to this ACE2 receptor here, gets taken in, the recipe is released, the virus is then made by the cell makes the virus, produces more particles, which then bud off. Okay, and that's how it works. Now it's more complicated than that, in that there is at least another receptor that's probably involved with this. But for the moment, let's just stick with this, if that's okay. Okay, so we really want a vaccine against this, and what we know is that the body has got very effective defense mechanisms, usually, to protect against infection. One of those mechanisms is called antibodies. And basically what happens is the antibodies are produced that then stick to the virus and then block it working. There is another part of the immune system called cytotoxic T cells, or let's call it cell-mediated immunity. And that's very good for killing cells that are infected. So there are two things to think about. Can you get antibodies, and can you get this cell-mediated immunity? So what's an effective immune response? This is important when you're producing a vaccine. So it looks like you need both cell-mediated and antibodies. That's what you need. You need both types of immunity. Right, what types of vaccines have we got? Well, the classic one we've got are what are called the whole organism vaccines. And this is polio. This is the two types of polio, the live and dead one. It's yellow fever. It's rabies. These are all whole organism vaccines. The toxoid vaccine, these are those where the organism produces a toxin, and you change that and you use that for your vaccine. Coronaviruses don't produce toxins, so we don't need to bother with that. We'll come to subunit in a minute. Let's look at the whole organism. Okay, so this is a Chinese vaccine. It's called BBIBP, but it's basically a dead whole virus vaccine. So this is like the injected form of polio. You get the virus, you kill it, you clean it, you put it into a syringe, and that's your vaccine. That's how you make killed polio. Now, where are they with this vaccine? Well, they've given it to monkeys. They've challenged the monkey with the vaccine. They've given the vaccine to the monkey. They've given other monkeys saline or placebo. So you're testing a vaccine, a possible vaccine, against a placebo. You then expose the monkey to the wild virus. Basically, what they've shown is that it seems to protect people, develop an immune response that protects them against SARS coronavirus 2. Okay. And they've shown that this vaccine produces antibodies in rats, mice, guinea pigs, rabbits, monkeys, and macaques. So it's a 2-dose schedule, and it looks like this whole virus killed vaccine works well to protect against SARS coronavirus 2. So this is one vaccine that will start to enter probably phase 2 and 3 studies shortly. Right, let's look at another vaccine. We've looked at whole organism, and we've got a whole virus vaccine that looks like it will currently work. By the way, just to say, there are 16 vaccines being tried in humans as we speak, and there are another 160 being developed. So that's 176. And it's probable, isn't it, that about between 5 and 10 of them are going to be successful. So you would think over the next 18 to 24 months, we should have 3, 4, 5, 6 vaccines that will be that should be protective for the global community. Right. Now, as somebody who's worked with vaccines, I look at this and say, this is very much like flu. Flu virus has a key on the outside that it uses to get into cells. So if I'm going to produce a vaccine against flu, I'm going to take the key and I'm going to use the key for my vaccine. Only the key, not the rest of it, just the key, because if I can then produce antibodies against the key, these antibodies will stick to it, block it, and stop it getting into cells. So I could do that using a whole organism, because the whole organism vaccine will produce antibodies against various parts of it. But it might be more efficient just to use the key as my vaccine, as we do with flu, and I will use a subunit vaccine. So this is what the key looks like. This is the spike key on the outside of the virus, and this is its genetic code. We know what we know from the Chinese what the genes are and what the instructions are to make this key. So this is one of these protein subunit vaccines, and it's made by a company called Novavex. Right. Now I'm just giving you this is just as an example. I would think the majority, certainly of medical practitioners in the room, have had a hepatitis B vaccine. And how you make hepatitis B is as follows. You take the virus, and you know that the surface protein on Hep B is its key. All organisms have keys that they can, all viruses have keys they can use to get into cells to replicate. So the key on the outside of this one is the surface protein. So what you do is you take that gene that tells the virus how to make the surface protein, you put that into a cell, a yeast cell, for example. The yeast cell is put into a tank to grow, and it produces lots and lots of hepatitis B antigen or key. You put that in your vial, and that's your vaccine. Okay? And that's how you make hepatitis B. Now, what Novavex have done is they've simply done the same thing. They've taken the gene and they have put it into some mammalian cells and produced a protein vaccine. It's quite an interesting vaccine. I'm not going to go into too much detail about it, but it has what is called an adjuvant that makes it work better. And it also is delivered through very, very small particles called nanoparticles, which means you can target it to the immune system. Now, what does it show? The evidence is that in animals, you're getting neutralizing antibodies. So it looks like this will produce antibodies. Now, if you remember earlier, I said you probably need both antibodies and cell-mediated immunity. So it's going to be interesting to see whether this vaccine works, because it's currently going into clinical trials, and it will be interesting to see, is it protective? Do you get both antibodies and cell-mediated immunity? The Chinese have done something similar. They've got a vaccine, it's Clover Biopharmaceuticals, and basically they've done the same thing. They've got the gene for the spike protein, and they've put it into a bacterial cell line, produced the spike protein, and that's their vaccine. And currently it's being tested on 90 adults and 60 elderly adults in Australia as part of an early study to see, does this vaccine provide antibodies and cell-mediated immunity? The big area, though, these days is what are called nucleic acid vaccines. So we've talked about the whole organism, and there are a number of those in clinical trials. We've talked about subunit, where you get your spike protein, you get the gene for the spike protein, you grow it in bacteria or mammalian cells, you produce the protein, and that's your vaccine. So we've got two lots of pretty straightforward vaccines. This is technology we're well used to. This is polio, yellow fever, rabies. This is hepatitis B. This is influenza. This is pneumococcal. This is Haemophilus influenza B, meningococcal vaccine. But it's straightforward, standard methodology. But this one is different. This is what is called nucleic acid. And basically, what do you do with this? Well, with a nucleic acid vaccine, what you do is you take the gene. So you take the gene as in here. This is the spike glycoprotein. So you take the same gene, but instead of putting it into bacteria or other cells and growing the protein and then injecting that into human beings, what you do is you inject the gene directly into human cells. So let's go back to our hepatitis B model, because it's really the same model. You take the gene for the surface antigen, you put that into a yeast cell or an insect cell, you produce the surface protein, you put the surface protein into a vaccine, you inject it, and it works. So what's this technology? What is this nucleic acid technology? Well, it's exactly the same, but instead of putting your gene for the surface protein into a yeast cell or an insect cell, you put it straight into a human being. So you miss out all this step. And by putting it into a human being, when the gene is transcribed, it produces the it produces the spike protein. The body recognizes the spike protein is foreign and produces antibodies against it. That's how it works. So this is the This is the genetic structure of SARS coronavirus. This is a spike protein here. So these are the amino acids that make up the gene. You take these amino acids, which is the gene, and that's your vaccine. Simple as that. Well, not quite as simple as that. So this is the Moderna vaccine. It's an mRNA vaccine. So what is it? Well, you use the gene, the mRNA gene, that codes for the spike protein. You take that gene and you basically deliver it into a human being. Now, the problem with giving mRNA is if you just inject mRNA on its own, it tends to get broken down very quickly in the body, and it wouldn't be an effective vaccine. So what you do is you put it in what is called a lipid nanoparticle. So basically, you put it in a small amount of fat. So this is what a nanoparticle looks like. Here's your fat. This is your nanoparticle, very small, less than 5 nanometers. And you can either put the spike protein gene on the outside, or you can put it on the inside. It depends what you're trying to do. Now, you then inject the particle, either this one or this one, and it's just like an ordinary injection. But what you also do is you put on the surface, you put certain molecules that will make this nanoparticle go to the cells of the immune system, where they will produce both, hopefully, antibodies and cell-mediated immune responses. So I hope that makes sense. You've got your gene that codes for the spike protein, and we've already looked at how you can use that in traditional subunit vaccines like Hep B. But you take the gene now, and you now put it into a nanoparticle, either on the surface or inside, and you then put targeting molecules around it so that it goes to the appropriate immune cells to lead to both, hopefully, an antibody and a cell-mediated immune response. And this is what happens. This is your nanoparticle with the gene on the outside. It gets taken up by cells that are important in initiating an immune response. It gets taken into the inside of the cell. It gets broken down, and the gene coding for the spike protein is presented to these cells here, and they produce cell-mediated immunity. And through a different pathway, they're presented to see these cells here that produces your antibodies. So the theory is that a vaccine with nanoparticles based on the spike protein will give you both cell-mediated and antibody adaptive responses. And that's what these studies are designed to do. Do they work? Well, this is Moderna's preliminary studies. They gave it to 18 to 55-year-olds, 45 people, and basically all participants produced antibodies by day 15. At day 43, 10 of the participants who'd received the biggest dose, there were 3 doses here, but 10 of the people who'd received the biggest dose showed levels of antibodies that significantly exceeded the levels seen in people who recovered from the illness. Now, we've got to be really careful here. These are preliminary studies. They tell us that this vaccine produces antibodies. They don't tell you it protects you. They simply say you can get antibodies that exceed the levels in convalescent sera. But one thing I would stress, we do not know currently what are the protective mechanisms against COVID-19? Is it antibody alone? Or is it antibody and T cells? Or is it T cells alone? We do not know. And this is where these different vaccine studies coming in are going to be really helpful. They're going to tell us what's going to work. Now, this is a really interesting vaccine. Not to say the others weren't, but this is an interesting one, This is the one that's been developed in Imperial in London. And I think this is also, they've got a collaborative arrangement with the Serum Institute of India. And what they've done is it's very much like the Moderna. It's very much like the Moderna. It's just basically an RNA spike glycoprotein. So it's the gene for this, just like the last one. But what they've done this time is they produce the vaccine and what they've done is there's your gene. And that's the gene for the spike protein. Now, they added other things to it, basically to make this multiply up. So instead of you just getting putting one gene in, because that's all this is, one RNA gene, by adding other bits to the vaccine, you make this multiply up and produce lots and lots and lots of RNA genes. So this is a really very interesting approach that by using this technology, you're putting in more RNA because it replicates. They also have a nanoparticle approach. So they're using nanoparticles to get it targeted, as we've just talked about. And apparently they're looking at having 20 million doses of vaccine available in December. And as I say, I think it's a collaborative arrangement with the Serum Institute of India. Okay, so let me move on a little bit. Yeah, you can see it's this lipid nanoparticle again. So let me just have a quick look. Shruti, you're telling me I've only got 5 minutes, but I was told I'd got 45 minutes, so I should have another 15 minutes left. So I'm hoping that's all right if I carry on. Is that okay?
It's fine. Yeah.
That shouldn't be a problem, doctor. You can please carry on.
Okay. Okay. So we've looked at whole organism. We've looked at subunit. And we've talked about the subunit that hepatitis B. We've started to look at nucleic acid. And what we've talked about is injecting the gene into human beings directly. And because when you inject them, if you inject the gene on its own, because that would be broken down very quickly, it wouldn't make a very effective vaccine. So basically you put it into a small lipid particle called a nanoparticle, and that enables the gene to be targeted to cells of the immune system. And we looked at a couple of those. Now, there's an alternative. One's got to think vaccines are really complicated. I was always told that vaccines were the most complex drugs you'll ever come across. And these are things to think about. But certainly in the administrative system, usually we inject vaccines. Okay, that's fine. In your children's lifetimes, they'll be using oral or edible vaccines. I'm quite sure about that, or transdermal vaccines, but we're not at that stage yet. But with delivery systems now, there are alternative ways of getting the vaccine in. As I've said, we can inject a nanoparticle directly. But there's another way of getting them in. And this is the last vaccines. I'll talk about 2 vaccines. But the interesting thing is, these are the ones I think that people are reasonably confident should be effective vaccines against COVID. We don't know, but we think so. So Let's go back. We've still got our spike glycoprotein. We know what the gene is that codes for it. And we think if we can produce an antibody against it, we can block it. But we probably also need cell-mediated immunity to kill any infected cell, because antibodies can't cross the cell membrane. So we think you've got to have antibodies and cell-mediated immunity. There's your spike protein again. Okay, we know its genetic structure. So what's the other way of getting it in? We've talked about getting it in through nanoparticles, and the nanoparticles target the cells of the adaptive system. Well, there's a different way of doing it. And what you can do is you can get it in through a virus. So this is an adenovirus, and most of us have had adenovirus infections. Probably 80% of us have had adenovirus infections as children, mostly asymptomatic. I think you can get a bit of a cold, but nothing else. So what do you do in this case? Well, do you remember with the mRNA vaccine, we had the gene? Okay, well, it's the same approach. This is the adenovirus genes. What you do is you cut out a bit of the adenovirus gene and you replace it with in this case, the spike glycoprotein. Okay, and so now you've got your adenovirus with the adenovirus genes, and we know the adenovirus isn't particularly pathogenic. It's now got a gene in it that codes for the spike glycoprotein. So as soon as this virus, you inject it, it gets taken up by cells, because all cells have ubiquitous receptors for this key. It gets taken into the cell, gets transcribed. It transcribes the gene for the spike glycoprotein. It produces the spike glycoprotein, which gets into the extracellular fluid, goes around the body, gets recognized by your adaptive immune system, and hopefully you then recognize it and produce antibodies and cell-mediated immunity. And this is the Johnson & Johnson vaccine. It's a non-replicating. So basically they've disabled this virus so it can't replicate. So you wouldn't expect it to cause any problems in terms of morbidity or anything like that. And you're using that very much like a nanoparticle. But instead of using a nanoparticle, we're using a viral vector. Now, some of you would and this is just how you produce it. I won't go into how you do it. But some of you would say, but hold on, if I've already been infected as a child and I've got antibodies against it, will that not block the vaccine? And the answer is probably yes. And that's what these studies are designed to find out. But what will probably happen is, yes, you will block it, but with the first dose, there's probably enough to prime the immune system, and then you'll give a second dose with a different vaccine vector. And that's probably what will happen with this vaccine. But what happens, you put the vector in, it gets taken up by your antigen cells, your antigen presenting cells, and it produces both cell-mediated immunity and cell-mediated immunity here and antibodies here. So pretty optimistic about that one. This is the one that the chief executive officer of the Indian Serum Institute is saying that they will be hopefully making it available in December this year. Now, they've taken a slightly different approach, slightly different, but not much. But instead of using a human adenovirus, they're using a chimpanzee adenovirus. Now, chimpanzee adenoviruses are not pathogenic in human beings. So it doesn't cause any problems. Only 1% of the population have got chimpanzee adenovirus antibodies. So you're not expecting the body's defense systems to block it. You put your spike protein in there, and then it's just the same as the previous one. And this is the University of Oxford AstraZeneca AZD1222 vaccine. And I would watch this one, folks, because I think it's the first one that's going to come out later on this year. And they showed it protected 6 monkeys from pneumonia, but there were 1 or 2 things about it that weren't quite what you wanted. But it may be that what we have to settle for is something like a flu vaccine, namely that it will stop you dying, it will make the disease less serious, but it doesn't necessarily protect everybody and block infection. But as somebody who's in an at-risk group for a coronavirus, if you were to offer me a vaccine that reduced my mortality risk, but didn't necessarily stop me spreading it, I'd be reasonably relaxed about that at this moment in time. So I'm going to finish with the last one now, just a couple more minutes, then we'll call it a day. Now, this is a replicating vector virus vaccine. So the previous 2 were non-replicating, okay? And they've got advantages in that they don't cause, they don't have the ability to cause disease because they're not replicating. So this is your Ebola vaccine. Now, this is the only vaccine, as far as I am aware, that is licensed. It's the only nucleic acid vaccine that has been licensed in America to protect against an infection. And what they've done is as follows. I'll just very briefly say it. So there's a virus called the vesicular stomatitis virus that causes disease in animals. It doesn't appear to cause disease in human beings. And when the Ebola outbreak was around, they did this. Now, if you think of this as the vesicular stomatitis virus, they took one of the genes out of it and replaced it with the Ebola spike protein, with the Ebola key. So again, very similar to what we're doing with COVID. And this is Merck doing it. Basically, what they're doing is they're taking this gene, and instead of using the Ebola glycoprotein, they simply replace it with the Covid19 gene. So this is a very similar technology to non-replicating, except these viruses replicate, but you replace the gene for Ebola with the gene for COVID-19. And the evidence is it's been very extensively tested in North America, Europe, and Africa. There were some side effects, sore arms, things like that, but it was safe and immunogenic in humans. So we know this platform works. And so you would expect that in human beings, if you replace it with the COVID-19, so you've just taken the gene out and replaced it with your COVID-19 gene, you would expect that it would be immunogenic and safe. But again, that's what these vaccine studies are about. Last slide, I think now. So where are we? Well, these are the different vaccines. We have got, these are vaccines that currently are in human trials. There are 16 of them. So there are 3 non-replicating viral vectors. So that's the Oxford one, for example. There are 4 whole virus inactivated. So that's the Chinese one, that's 7. There are 3 protein ones that we talked about. That's 10. And there are 6 of the spike protein gene genetically engineered vaccines. These are currently in human trials at the moment. There are another 160 that are different types of these. They'll all be by injection. there'll be nanoparticles or non-replicating viral vector. There is one replicating viral vector vaccine, which is the vesicular stomatitis, but that's not yet in human trials. We talked about nanoparticles, so that's where we currently are, folks. That's it. So what I hope I've done is I hope I've described something around how vaccines work. I've described something around the epidemiology of COVID-19, and I've looked at some of the examples of vaccines that are currently being trialed in human beings, and which almost certainly will be available at the end of this year, which will be remarkable. That means we'll have a vaccine that's potentially testable in 12 months, because it was only identified in January. So we'll have a vaccine potentially in December. And when you think that polio took, 50 years, flu took 30 years, diphtheria took about 40 years, quite remarkable where we're at. Anyway, that's it, folks. Hope that was okay.
Yes, that was a great insight to the whole of this virus and the formulation of what are the modes of vaccine which can come in future, and when it can probably come to us. That's a really wonderful eye-opener for David for that excellent talk. I have a few questions. One, Mr. Kevin Mark wants to know.
Shivshankar, 1 second.
Yeah.
I think on the YouTube live channel, we are just lagging behind few minutes.
Yeah, that's okay. They gradually, they will be 1 minute behind us. That's okay.
Yes, doctor, there'll be a delay,
Yeah.
but then there is no problem.
That's correct. Yeah.
Okay. In
There
terms
is
of catching the questions.
Question from Mr. Kevin Mark. How these vaccines work when the virus is mutating? That's the first question.
Okay, so it's a really interesting question, that, and it was good talking to you earlier, Dr. Shivshankar, because you were telling me about 40 or 50 different strains in India, which may be associated with different pathogenicity or at least different mortality rates. I think I didn't actually show it because I was slightly worried I was running out of time, but can I just try and show you one thing? With one of the vaccines that I looked at, one of the vaccines, they were looking at, it's an inactivated vaccine, so it's one of the early ones. And if you look here,
like the one, the polio, inactivated.
Yeah, this is the inactivated one, exactly right, yeah. Yeah, you picked it up. So I was looking at the phylogenetic tree, and this is the phylogenetic tree here. Okay, so these are the strains that have been identified so far, and you can see that there've been 3 strains. Now, by this, they're looking at significant mutation. All coronaviruses seem to change an amino acid every few replications. But that's not a significant change, or it doesn't appear to be a significant change. So here you've got 3 strains. You've got a CQ01, QD01, and HB02 here, and they're here on the phylogenetic tree. Now, when you actually look at them, there is 100% homology against the spike glycoprotein. So although you seem to get amino acid variations, that doesn't appear to be significant in terms of developing a protective immune response. But we have to wait and see. This is what research is going to be about. We're going to watch over the next 1 month, 6 months, 12 months, 2, 3, 4 years, and see, because the point is very valuable. Is it going to be like flu? Are we going to finish up giving 4 flu vaccines every year because we have an endemically transmitted virus that mutates?
Yes. Okay. The next question is, since there are so many strains of this viruses in the air, if I get this disease once, what is the chances of getting permanent immunity against that, or whether I'm going to get reinfected with some other strain in subsequent days?
In a way, that's very similar to the first question. It really depends on what's the nature of immunity. If the immune response, which I suspect is going to be both antibody and cell-mediated, if that provides protection against minimal mutation of the spike glycoprotein, then you would expect immunity to be protective. Okay. But there is a problem to that. It's the duration of immunity. Is the duration of, so for example, if I give you a polio vaccine and I give you 3 doses, I'd expect you to be immune probably for the most of your life. Yeah. Now with coronaviruses, with the common cold ones, immunity lasts 3 or 4 months. With SARS and MERS, it appears that immunity lasts about 8 years, but we really don't know. And I don't think people will know what the immunity is going forward with COVID-19 unless we do the studies. And you'll find that in a year's time, we'll know how long immunity to natural infection is, how long immunity is to the different infections and different vaccines. And that will build up as knowledge increases.
Yes. So this is a RNA type of virus. So even HIV virus is also a RNA type of virus. Over the last 36 years, we are not having a vaccine against HIV. Then how do we predict that we are going to have vaccine by the end of this year?
Well, I think it's quite easy to predict we're going to get a vaccine because we know that the manufacturers are making them. And the preliminary results from, for example, AstraZeneca, the chimpanzee vaccine, we know that it protects, we know it prevents macaque monkeys getting pneumonia. That's been shown. So what's happening here is because this is a global, potentially a global catastrophe, the clinical trials are running in parallel with producing the vaccine. So again, I'll come back to the Indian Serum Institute and the CEO. And what somebody asked the CEO, why are you producing millions of doses of vaccines that you don't know if they're going to work or not? And his response was, well, we're a private company, we can potentially take the hits, but the preliminary results look good or look effective. What we'll do is we'll produce the vaccine, and almost certainly they will make it available as part of a phase 3 study. So it won't be a licensed vaccine. So this isn't the licensed Hep B vaccine you and I have had, Dr. Shushankar. It's not going to be that way. What it will be is it will be a phase 3 vaccine that isn't licensed, but we'll say, let's make this available under emergency concern, and because of the pandemic, we will trial it out as we're delivering it. And who do we give it to? Well, healthcare workers, because we are the first line. Who else? Well, people with chronic lung, chronic heart, chronic kidney, people at risk. And also people like me who are getting a bit on in years, or very old people who we know in my country, people over the age of 85 have a mortality rate of, I think, about 35% if they get it. Now, if I was 85 and I knew I'd got a 1 in 3 chance of dying if I get it, I would be very keen, I think, to have a vaccine. Because the point about it is, in my country, it's thought that by the end of the first wave, 10%, perhaps 12% of people will be immune. That means 85% are not immune. And if the disease is reintroduced, you will get more cases transmitted.
Yeah. Why this disease is playing different roles in different countries? Because we read in papers that Italy had very high mortality, then Spain had very high mortality, even England UK had high mortality when compared to other nations like Germany, which had the least mortality. Even in India, we have got a national mortality rate of about 3% of the total number of cases which have been detected. Why there is difference, so much of difference from 30% to 3%? That's very...
Well, we talked about this right at the beginning, and I thought your mortality rate was unbelievably good.
Yes.
Really unbelievably good.
And for your information, India has more than 1/5 of the world's population, almost about 160. Europe, America, South America, Australia, New Zealand, everything put together. We have got that much of population in India.
Yeah. So what do we know about mortality from this disease? Well, we know that men have a worse mortality than women. We know that. Well, certainly in the UK, it's 2.8% versus 1.7%. We know that the mortality is worse in older people than younger people. We know that. In my country, certainly, Black, Asian and minority ethnic populations have a higher mortality. We know that certain groups, occupational groups, have a higher mortality, classically healthcare workers. So there are some factors there. And you may, for example, expect that if in a population with a predominantly younger age structure, you would expect perhaps lower mortality than in a country like mine, where the age distribution is much towards the upper end. You mentioned Germany. You could have mentioned, I think, South Korea, Singapore. And one of the things, there are a couple of things I think that really good to see what the Chinese did. The Chinese effectively, once this outbreak had started, the Chinese effectively locked down cities. You were not allowed to travel. They then developed programs in each city where they checked people's temperatures, they identified potential cases, they isolated, quarantined them, and they basically stopped the disease spreading within the particular city. Yeah? And there was a similar study from a town in Italy where there were about 3,000 people in the population. They screened everybody in the population with a virus test, and they found something like, I think, about 70 people, 90 people who were positive. Half of them were asymptomatic. They isolated everybody for 2 weeks, and the virus died out. And there were very, very few cases subsequently. I think one of the things that is really helpful very early on in an outbreak is if you close down people moving around, if you close down, for example, air travel very, very, very early on, not several weeks into it, you can stop disease spread. If you identify cases very aggressively early on, you test them, you isolate them, you quarantine them, you advise your population about all the appropriate measures that everyone's doing, social distancing, masks, etc., etc., you can control it. And I think in Germany, they had a testing system that seemed to be available several weeks earlier than some other European countries, and they therefore, I think, had a better experience. The Italians may well have been slightly later than Germany in implementing the control measures, facilitating wider travel. Does that make sense? Not wider travel, wider transmission. Does that make sense?
That makes sense, but is it lockdown is just a wishful thinking that we are delaying the disaster to happen, and also to get ourselves prepared, enhancing our ICU beds and medical facility, and waiting for a vaccine to arrive in the market? Because if you are not infected right now, but whenever the country is opening up, we are going to get this virus from somewhere. Isn't it that is the nature of this virus, and we are just locking down only for the postponing the disaster to happen? That's what you feel?
I think that's a really valid point. I think one of the things I'd say is that by locking down, as you said, and I completely agree with you, it gives you the opportunity to develop your intensive care facilities, including ventilators, etc. And it enables you to perhaps start to stockpile things like remdesivir, start to look at things like convalescent serum that we've talked about, plus all the other drugs that people are starting to look at. So, for example, proning, turning patients on their face seems to improve mortality. Dexamethasone improves mortality. So we're getting better at managing people with COVID-19. But you're right, potentially, I've just said it to you, 85% of the population in the UK will be susceptible.
Yes.
But this is where the vaccine comes in.
Yes.
You know, we're hopefully developing a vaccine that in 6 months' time will start to provide protection, even though it is being given as a phase 3 study. But otherwise, all that's going to happen if we say, well, let it just happen, well, that's going to, a lot of people are going to die as a result of that.
Yeah, that's correct. Why there is a problem with the disease affecting more of old age people? Is it due to the T cells or the thymus cells, or it's the general immunity or anything else related? Because we see some of the very old people recovering without any problem, and some of the very young patients having all the viral load and going into all the problems.
Yeah.
So why this happens?
I wish I knew. I mean, I can give you the standard answers, which are that the older you get, the more morbidity you've got. You've got cardiac morbidity, you've got impaired renal function, you've got respiratory morbidity, and you mentioned those, and we know that there is increased morbidity and mortality with these conditions, and you see that with influenza, and my suspicion is it's just the same with COVID-19. So I think that's one of the things. But you also mentioned the other problem, which is waning immunity. And I'm sure that the patients you see, you'll see some 80-year-olds who are biologically very, very fit. But equally, you'll see some 60-year-olds who are biologically very impaired.
Physiologically, they're old. Yeah.
Yeah. And you're probably thinking, oh, if I operate on this particular individual, they're going to have an increased mortality or morbidity at least, because they've got all these underlying problems. Now, we know, for example, that antibody response and cell-mediated immune response, it gets worse as you get older. It's called immunosenescence. And for example, my immune response to COVID-19 is going to be worse than yours. You're younger than me. I'm more immune senescent than you are. I can't recognize one aspect of immune senescence is my neutrophils. Immune senescence has got two components: the innate and adaptive system. In the innate system, my neutrophils don't work as well. In the adaptive system, I can't produce antibodies against new pathogens as well as a younger person. Okay. So yeah. I think you're seeing a combination of things there, and my guess is there's also a genetic element to this. When I say genetic element, I don't really know what I mean, but there's some people who are, as I say, very fit.
Correct. Okay. There is a repetition of similar question. Does the new corona vaccine would be sufficient to combat against different strains and mutations of the coronavirus? And also, would this be globally accepted? I think you have answered this question earlier. Yeah.
Yeah, I think we've answered that.
Kavya Jagdish wants to know, is this vaccine immunogen which you are talking, are these Chinese vaccines? One person wants to know. Are you are talking about what is being done in general or done in UK?
I'm sorry, I should have made that very clear. The 16 vaccines that are under human trials are all global, excluding China. So the Chinese vaccines, there are a couple of live vaccines they've got. I think they've got three whole virus inactivated. I'm quite sure they've got some of the non-replicating viral vector vaccines, but it's less easy to get all the data for those. And the World Health Organization, which pulls together the sort of clinical trials around coronavirus, have focused largely on Western vaccines. These are not UK vaccines. We in many ways, what's happened to the UK is, anyway, it doesn't matter. You are now a much bigger player in vaccines.
Yeah. Okay. At the outset only, I will apologize because it's difficult for me to pronounce the name. It's something like Gnanam Hugh Kotnis. The question is this vaccine suggested is both for child as well as adult?
Don't know at the moment. My guess is what will happen is we will start with the elderly, then the 65 and above, the under 65 at risk, and health workers and other essential workers. And I think it will be very much a phased program. The disease in children, not exclusively, but the disease in children tends to be a little milder, and the morbidity doesn't appear to be as in older people. And my expectation is that you will test the vaccine in older people first.
Okay. One Mr. Sadiq Khwaja wants to know, do you think there is different phenotypes of coronavirus which is confusing the picture and producing asymptomatic to no anosmia only to severe end?
The evidence that I've seen, I'm not a virologist, but the evidence I've seen would suggest that the changes in the virus at the moment do not appear to be leading to significant changes in morbidity and mortality. That's what my virology colleagues tell me. But this is, if you think about it, we first picked up these cases in mid-December with people coming in with atypical pneumonia who worked at the Wuhan sea market. And we're now in 4th of July, which is what, 6.5 months later. We know a huge amount about the virus. We've got potentially 16 vaccines that we're starting to test. And we're looking at a number of treatments, some of which are clearly effective. Dexamethasone works for some aspects of the exaggerated immune response that you get in the second plus week. We know that remdesivir reduces mortality. So in 6 months, 6.5 months, a lot has been learned by scientists and doctors throughout the world. It's remarkable what's been achieved. But there are some things, well, there's certainly some things I don't know. So
yes, there is one more question from Kavya Jagdish. At the beginning only, I'll tell that we are already overshot the time almost by 25 minutes, but we'll take a quick question. Kavya Jagdish wants to know, can these immunogen vaccines can be used in any patients, irrespective of their health condition like diabetic, renal problem, anything?
Yeah, good question. And this is partly what these studies are designed. If you think of somebody who's immunocompromised for whatever reason, and that could be, could have a primary immune deficiency disorder, they could have a secondary one associated with treatment, they could have it associated with disease. So people who've got diabetes, renal impairment tend to be immunocompromised. Now, with those, you would probably tend to use an inactivated vaccine rather than a live attenuated vaccine. You may use multiple doses, and you may use a larger dose. And that's all what these studies are designed to show. What's the best vaccine or vaccines in different population groups? I think the point I would make is we do not have a licensed vaccine. What we've got are 16 potentially very interesting vaccines. And over the next 6, 9, 12 months, perhaps 18 months, those vaccines will be identified as which work better and which don't work as well, which work better in certain groups compared with others. What's the best dose? Do you use more than one schedule? Do you use more than one dose, or do you need 2 or 3 doses? Do you need it every year? These are things that will be being understood with the trials that are currently being done, but we don't have a licensed vaccine at this moment in time.
Yes.
I'd be saying to you, really, really be careful, folks. If you're in a community where SARS coronavirus 2 is transmitting, make sure you follow all the guidance that people talk about: hand washing, masks, social distancing, shielding for older people. You've really got to do that.
Yeah. So with the current phase 3 trials going on, how early do we predict we can get? Because the Indian government has set a limit of 15th of August. Yesterday only it has come in the newspaper that we should have a vaccine by 15th of August. Is it we can fast track this test so quickly, or it will take you? Do you feel that it will take much more longer time?
I don't know enough about the Indian vaccine scene to answer that question. Are they saying that by the 15th of August you will have a licensed vaccine, or are they saying there will be a vaccine available which can be tested in a phase 3 study, and at the same time you could give it to high-risk individuals? Because I think that's what is likely to happen, that you'll get a vaccine which you think, this is really promising. The Oxford vaccine looks promising. The Imperial vaccine looks promising. 3 of the American ones look promising. I don't know about the Chinese ones, but they look promising. Now, they're all multinational. As I say, I think 3 of them are being delivered through the Serum Institute of India. Now, if they're at the stage where they're looking very promising, we can try them out in at-risk groups, then in which case that could be that there'd be a vaccine available August the 15th. It wouldn't be licensed. You couldn't give it to everybody, but you might make it available because of the global pandemic to people at higher risk. And you would then see how it works and how protective it is. I'd be very surprised if you've got a licensed vaccine on August the 15th. I'll be completely honest.
That's fine. So I have taken all the questions which have come online on the YouTube. So thank you very much. It was really a fresh insight to the way the things are working towards the corona. We were all very much worried 6 months back. Now we are much more lenient. We know little more, as you said. As the time is advancing, we are knowing more better things, and probably we are getting ourselves equipped with all the things so that the mortality is reducing. So hopefully the vaccine also comes in our armamentarium very shortly, so that lots and lots of lives will be saved in future. I'll hand over the proceedings to Dr. Shashidhara for further things.
Thank you. Thank you. First and foremost thing is David, Professor Baxter, for doing this on a Saturday. It's a weekend in UK, and spending time, and we've overrun by nearly 25 minutes. Thank you very much for that. Every time I hear David, it's probably, I keep hearing about, I learn something new every time. And thank you. I hope this is useful for them. And other important thing is, I only asked Dr. Shivshankar to chair this session because the other person wasn't available. And I think there's only about 2 hours ago, I asked him, can you please share this session, because somebody else who was supposed to chair couldn't make it. Shivshankar, thank you very much. Thank you very much for chairing the session, and also gathering all the questions and putting it across to David and get the appropriate answers. And the most important thing is, obviously, thank you very much for all the people who are coming to watch this on the YouTube. I hope we have answered some of these questions. These kind of sessions will carry on, and we are going to have some more sessions on the weekend next week. I believe Dr. Devi Shetty is doing a session on February. Is it Friday next week?
Yes, doctor, that's right. It's Friday.
Yeah, Friday, and similarly, we are going to be continue to do this. In addition to this, we do have a survey going on from Ms. Saidi Khaja, who asked a question. She is doing a study about taste and smell. There's a survey going on which is going to be circulated to all the healthcare workers in India in terms of gathering some data from India as well. It's a global study. As you know, that Professor Nirmal Kumar, President ENT UK, was involved with big fact-finding mission in terms of loss of taste and loss of smell being the important symptom for COVID-19. There's a survey about that. And thank you very much all the people who signed on in the YouTube. And lastly, I also want to thank Shruti and Prajwal for setting up the link both on the Zoom as well as a YouTube. And without you guys, this would not have been possible. And once again, this came from the Global Training Education Center Global Medical Program. Thank you. Thank you, everyone.
Well, that was a very informative session, and I thank you, Professor David Baxter, for highlighting the situation, the vaccine classification, the information about the vaccine to be available soon. That's a relief. Thank you, Dr. Shiv Shankar, for the wonderful questions, and thank you, Dr. Shashidhara MK, for taking time and doing this for us. We are receiving messages on how wonderful this session was. Step One is honored to have you here, and yes, we do look forward to more such sessions. I also take this opportunity to thank all the doctors who are volunteering and the volunteers for your wonderful service. Remember, until the vaccine becomes available, follow social distancing and personal hygiene measures to keep your loved ones and yourself safe. Stay tuned for more interesting sessions, and don't forget to attend the 10th July webinar about virtual healthcare by the very famous Dr. Devi Shetty, Chairman of Narayana Hridayalaya. Thank you again. Stay tuned. Bye.
Bye.
Once
Thank you.
stop live streaming, we'll just check that for a minute.
Yeah.
You have stopped, Shruti?
Yes.
Yes. Okay.
Just one more.